scientists and more than 18,000 graduates contributing to agricultural development. UPCA, now College of Agriculture and Food Science, or CAFS, has been continuously contributing to nation building through its instruction, research, and extension function. A mere 10 years after establishment, the college was able to start breeding works with the production of sugarcane varieties with high productivity and resistance to Fiji and mosaic disease. In the 1920s, the importance of agriculture was intensified and UPCA was recognized as neighboring countries began sending their students to Los Baños because of the high quality of education. In the 1930s, breeds with superior characteristics such as the Burkhala pig, Los Baños Cantonese chicken, and Philippine cattle were developed. The 1960s was a fruitful decade for research. Makapuno, a coconut cultivar often made into a dessert, was pioneered and perfected. Meanwhile, the Dairy Training and Research Institute was the first in the country to freeze buffalo semen, paving the way for artificial insemination of local carabaos. Along with the establishment of UPLB in the 70s, the college received the prestigious Ramon Magsaysay Award for International Understanding, Asia's counterpart for the Nobel Peace Prize. In the same decade, the C4 plants were developed. The soil test kits were produced in support of the Masagana 99 program, and the mango induction technique was introduced. Continuing the production of quality research outputs in the 1980s, techniques to minimize post-harvest losses, the accelerated fish sauce manufacturing process, and the rapid test kit for the detection of pesticide residue on vegetables were developed. The 1990s saw many new varieties across species or crop types, including Sinta Papaya, the first Philippine-bred hybrid, UPLB Gold, a durian variety, and Doña Amelita, a musaenda named after the wife of former President Fidel Ramos. The award-winning National Corn-Based Farmer Scientist RDE Training Program, or FSTP, was also established around this time and still remains as one of the university's primary extension programs. In the field of instruction, the book titled Integrating Sustainable Agriculture in the BSA Curriculum, the UPLB College of Agriculture Experience, was published in 1999. At present, the College of Agriculture and Food Science, or CAFS, continues to be the premier institution of higher learning in agriculture and food science, and remains committed to nation building through its instruction, research, and extension programs. In order to produce the best graduates, all of our courses are outcomes-based education, and continuous improvement of the curriculum is being practiced to meet the challenges of the 21st century. As testament of excellence, CAFS is constantly recognized by different organizations. In research, we continue to implement relevant studies with focus on the 12 priority commodities to address food security. Moreover, technologies are constantly developed to aid in responsible food production. Supplementing these are extension activities to disseminate the knowledge generated to improve food productivity and livelihood of the people. We believe that present technologies will future-proof our food systems. Some of our notable developments are IDTech, Coraginam, Plant Health and Pest Clinic, Pest Management Tools and Techniques, Smarter Pest Identification Technology or SPITech, SNAP hydroponics, drone technology, rapid immunofilter assay for virus indexing, nanofertilizers Fertigrow, Corbogrow, nanobiosensors, and biochar. We at the College of Agriculture and Food Science will continue to promote and nurture a culture with innovative yet conscientious minds and cutting-edge skills to lead and serve the country and the region. We are one college with one goal towards sustainable food systems.
Good morning to everyone. Welcome to the third episode of Plant Health webinar series brought to you by College of Agriculture and Food Science of the UP Los Baños. For today, we will be talking about push-pull technology and companion planting, a dual strategy for insect pest management. This episode is co-hosted by the Institute of Crop Science and the Institute of Food Science and Technology. I am Dara Maria Fabra Realin, Assistant Professor from the Institute of Crop Science, and I am your host for today. We also have with us is Dr. Florencio C. Regino Jr. from the Institute of Food Science and Technology, our Q&A moderator. Just a few reminders for all of us for a smooth flow of our webinar. May we request all our panelists to keep your microphones muted at all times, except if called or requested. For our Zoom participants, during the open forum, use the Q&A button or raise your hand to be recognized. For our Facebook and YouTube participants, on the other hand, kindly use the comment section for your questions. On September 28, we launched the Plant Health webinar series. For the first episode, we had Dr. David Shapiro Ilan, who presented to us the approaches to improving entomopathogenic nematodes as biocontrol agents. Then on the second episode, we had Dr. Isa Nurkumar, where he presented the new association between invasive pest Spodoptera frugiperda and native parasitoid species in the special region of Yogyakarta, Indonesia. Now, to formally open the program of our third episode, I would like to call on the Director of the Institute of Crop Science, College of Agriculture and Food Science, UP Los Baños, Dr. Eureka Teresa M. Ocampo, to give us her opening remarks.
Hello, good morning, ladies and gentlemen. I'd like to welcome you to the third webinar on the series of the Plant Health webinar series. So the first episode, as already mentioned by Ms. Realin, was approaches to improving entomopathogenic nematodes as biocontrol agents, followed by association between invasive pests and parasitoid species in the special region of Yogyakarta. And for our third episode, we have sort of a conglomeration of the first two the concepts presented in the first two webinars. And this is on the push-pull technology, which utilizes crop production practices and biocontrol agents towards better production, leading to sustainable agriculture practices. So we are really proud that we were able to invite one of our very own for today. And we look forward to her talk, and we hope that we learn a lot of things that or concepts that we can use in teaching, and also in which we can also practice when we are working towards better sustainable systems. So I would like to welcome our guest speaker for today. She will be introduced later. And thank you everyone for coming. I can see that we have a very good attendance this morning. Thank you for coming. Welcome to the webinar.
Thank you very much, Dr. Weng Ocampo. At this point, Mary requests everyone to turn on your videos and give us your best smile. For our group photo, may I request the assistance of the CAPS technical team? Okay po. Sige po. First page. Okay.
Okay po, ready? 1, 2, 3. One more po. 1, 2, 3. Isa pa po, nagbubukas pa po atang camera. Okay po, ready? 1, 2, 3. Okay po.
Thank you, Ma'am Kat, for the assistance. Thank you very much, everyone. So now let us proceed to the introduction of our speaker for today. Our speaker for today is Dr. Jesusa C. Legaspi. Currently, she is a courtesy adjunct associate professor and former co-director of the Center for Biological Control at Florida A&M University in Tallahassee, Florida, USA. She recently retired as research entomologist in the U.S. Department of Agriculture, Agricultural Research Service, or Center for Medical, Agricultural, and Veterinary Entomology. Dr. Legaspi also obtained her PhD in entomology at Purdue University, Indiana, USA, took her master's in applied entomology at the University of Newcastle upon Tyne, UK, and BS Biology with a minor in zoology at the University of the Philippines, Los Baños. Her previous research involved the use of sustainable technologies and natural control agents of insect pests in organic crops. Dr. Legaspi's research work experience included working in international, national, federal agencies, and state universities. To give us the webinar titled Push-Pull Technology and Companion Planting, a Dual Strategy for Insect Pest Management, we are very honored to have her as a speaker today. Ladies and gentlemen, let us all welcome Dr. Jesusa C. Legaspi.
Thank you so much for the introduction. I'm very honored to have been invited to this plant health seminar series. And it's a good chance, actually, since, as mentioned, I recently retired. So it's a nice way to pretty much be able to present my research work towards the end of my career. And let me go ahead and I'd like to also thank the organizers of this seminar, and for those of you who are attending this seminar this morning. It's evening here in the US, but that works well for me. And let me go ahead and share my screen and start my seminar. I'll start from the beginning. All right, as was mentioned, this is the topic for today. And I will go a little bit, although I've been introduced with a little bit of my background, I thought I would still give a little more detail about my research activities, as well as some academic activities as well, since I am a former research entomologist for U.S. Department of Agriculture, as also adjunct associate professor at Florida A&M University. And in this capacity, I've been able to train the students, undergraduate, master's, and PhD students in research. And I would occasionally give seminars of my research when invited. So as mentioned earlier, I graduated with a BS Biology at University of the Philippines in Los Baños. So I really appreciated that video earlier. It took me back, in the mid-70s when I started my studies there. So it was just so nice to be able to see the changes that have occurred. And I went ahead after my receiving my bachelor's. I was hired by actually a government agency to work for actually an international organization, but I'm sort of getting ahead of myself. So I continue on with the master's in applied entomology at the University of Newcastle upon Tyne under a British Council scholarship. And then went back to the Philippines to work at International Rice Research. And then I went to the US for my PhD in entomology at Purdue University. And I did, as I mentioned, started out as a research assistant for the Philippine Council for Agriculture, Aquatic and Natural Resources, Research and Development. At that time, that was the name. And then also it was a collaborative project with the Southeast Asian Regional Center for Graduate Study and Research in Agriculture, based at the International Rice Research Institute. And because of that experience, I was then sent to the UK to do my master's. And when I came back after that, then I worked for IRRI and did a couple of years work as a research assistant. Then I went to Purdue University. And after I graduated with my PhD degree, then I did a short postdoctoral research at the USDA Agriculture Research Service in Weslaco, Texas, and then went on to be a faculty at the as assistant and associate professor at Texas A&M University. This was an off-campus institute, so I was based in Weslaco, Texas. And I suppose for the past 23 years, I've been at the Florida A&M University campus as a research entomologist for USDA. And I was co-director of the Center for Biological Control in Tallahassee, Florida. And in this capacity right now, although I'm retired, I continue as an adjunct associate professor for Florida A&M University. So I'm in the committees for in several master's students and graduate committee. In terms, I'm sorry, I went back. Research activities, as I mentioned before, here are just some photos, which I know I'm dating myself now, but I did start as a, I'm sorry, I had to, I pressed the wrong button. I did start working for a British couple that was based at the International Rice Research and we were doing research on the brown plant hopper, which was is still a major pest in the Philippines in rice. Then when I did my master's, I did work on potatoes at the Rothamsted Research Experiment Station in Harpenden, England. So these are just pictures of some of my research activities. But today I will mainly talk about my work on the sweet potato whitefly, which is a major insect pest in the US as well as other parts of the globe, and is also becoming a pest actually in the Philippines. I will mainly talk about this cultural control method of push-pull technology and companion planting, although I will show later that I've done more pest management research studies on this pest, which is really spreading around the world now. So this is a picture of the adult on the right. It's very, very small and hard to see, but this is the kind of population numbers that you would see. This is underneath the leaf that causes a lot of the damage. And as you can see in the lower left, the damage caused by this insect in just a couple of weeks, you could end up with this kind of damage. And on the right is called the silvering of the plant because of the virus caused by this insect. So just a little biology of this insect. This shows a picture of a nymph or an immature sweet potato whitefly. It's also sometimes called silver leaf whitefly. So sometimes you may see that acronym, like in this case. And this is the adult, and next to the adult are the eggs that they produce. A little bit of the biology. It takes about, from egg to adult stage, takes about 18 to 30 days at 25 degrees centigrade temperature. And the adults can live from 10 to 22 days. They're very productive in the sense that they could lay up to 3,000 eggs per female, and that they do attack many plant species. As you can see here, 420 plant species from 74 families. And the main problem with this insect is not so much the physical damage, but the transmission of viruses, which causes the death of the plants. So they can transmit 20 viruses, which leads to about 40 crop diseases. Again, these are just pictures of the life cycle of the Bemisia tabaci from egg, from the left. In just a few days, it becomes a first instar, which moves, but then when they become second instars, they are practically immobile on the leaf, and then they get to the third instar and the pupal stage as shown in the lower pictures, and then the adults. So it's about about 2 weeks to 3 weeks for the whole cycle of the insect. And as I mentioned, I've been working on other methods aside from the cultural control I mentioned. I've been working on, or I worked on biological control using native and imported natural enemies, as well as use of augmentative biocontrol. I've tested different biorationals, pesticides, oils, and those type of management for the sweet potato whitefly. I've also done work on entomopathogens, for example, fungi, as well as nematodes, and also the use of conservation. So when you integrate all these various techniques, that's what's called the integrated pest management against the target pest. So when I first started working on this pest, sometime in the early 90s, it was already spreading around the world, and this was the distribution of these Bemisia tabaci. You can see the red dots here on the map was pretty much the distribution at that particular time in the early 90s, but a more the most recent I could find was this distribution in March of 2018. So you could see more of these red dots here spread all around the world, and that may be due to the climate change, the warm weather we're experiencing, as well as the global trade. So my experience in Florida is pretty much my topic for today. And Florida produces or is second in the US for production of vegetables, about 1.3 billion and US dollars, and grows about 250,000 acres of these vegetables. And it's also ranked one of the top 3 for tomatoes, bell peppers, snap beans, squash, sweet corn, and others that you can see here on the slide. So to be able to conduct this management strategy of integrated pest management, or whitefly, I collaborated with various research institutes, as well as universities, for example, Texas A&M University, Florida A&M University, as well as stakeholders such as the Florida Organic Growers Incorporated and the Red Hills Small Farm Alliance. I worked closely with, I'm sorry, I may have missed some other agencies. I worked closely with organic growers in North Florida. Other universities I worked with are University of Georgia and University of Florida. So while when I moved to Florida, I pretty much worked on IPM using spatial and landscape ecology, research, biological control of invasive pests, and looking at the basic biology, growth physiology, field ecology of these pests in vegetables and horticultural crops. So this is just another picture of the whitefly adults and how you see them underneath the leaf. And my interest on using push-pull started when I was looking for plants to use for my field study. And I noticed in the nursery when I was purchasing plants to start as transplants, I noticed that the mustard plants next to cabbage or other vegetable transplants had very, very low whitefly numbers compared to the crops such as cucumber, cantaloupe, and squash. So I was wondering if there was something in the mustard that may be a repellent against this whitefly, or whether it's just not a good host for this whitefly. So I was interested in looking at that. There was not much literature about it. So I started looking at the use of these companion crops with repellent or masking volatiles as a potential crop protection method. I may have gotten stuck. Let me see. I'm not able to move this slide. I wonder if you're able to move it from there. Okay, now that's better. So this use of repellent crops has been incorporated in the concept of the association resistance, or as the push component in push-pull pest control strategies. I think there may be a delay with these slide. I apologize. Do you see any slides there that are changing? I'm sorry to ask.
Ma'am, yung slide po ay nasa evaluation of mustard plants po. Tas
Okay,
dun pa din po.
okay, all right. I think there is some sort of delay from my end. So I went ahead and did some field experiments using combining crops with non-host masking odors. Previous studies have shown mixed results. I wanted to go ahead and test the use of these companion crops or repellent plants. So the objective of this study was to evaluate the effect of the mustard, the variety giant red mustard plant, Brassica juncea, and commercial products to control Bemisia tabaci. I used no choice and choice tests. I also did a greenhouse study and a field test of the oviposition or egg production of Bemisia on other vegetable crops. So for the no choice and choice study, I released whiteflies into cages, as you can see here in the picture, with potted squash plants, and I sprayed different oils on the plant with 2% tween as a spreader. And so I used mustard oil, garlic oil, horticultural oil, hot pepper wax, and water was used as the control. So the no choice test setup is shown here on the left picture, and the choice test is shown on the right, where I had a plexiglass cage showing the two plants. One was treated and the other was untreated. So we released 20 Bemisia tabaci females in each of the pots or the cage, and after 24 hours we went ahead and counted the eggs that were laid by these adults. And after the 24-hour period, then we counted the eggs underneath the leaf, and this is how it looks like underneath that leaf. So in terms of the results from this study, what we found were that the control had the highest numbers of whitefly eggs, followed by the hot pepper treatment, the petroleum, the garlic, and the mustard. So these were all, the treatments were all significantly different than the control, which was the water spray. So you will see mustard here had the lowest numbers of Bemisia tabaci eggs. And then in terms of the choice test, where we presented the Bemisia with a choice of either a control where it was just sprayed with water or a mustard oil, you can see the difference here that there were more Bemisia eggs laid on the water control versus the ones that were treated with mustard oil, and that was had the significantly different numbers. But although the ones that were sprayed with garlic, hot pepper wax, and petroleum were not significantly different. So to summarize, there were lower numbers of whitefly eggs in plants which were sprayed with mustard oil and other oils compared to water control. So going now to a greenhouse experiment that I did, I looked at plant pair treatments per pot. For example, I had a pot that was planted with a colored next to another colored plant in the same pot, or a pot that had a mustard and a colored next to each other, or the third treatment where I just had mustard and mustard in the same pot. So we then infested these treated pots with sweet potato whitefly by having potted plants with plenty of whitefly. And we did 2 trials with 48 leaves per treatment. So 6 days after we infested these treatments with the whitefly, we then collected a single leaf per pot and counted the numbers of eggs and nymphs underneath the leaf. I will go on to the next experiment method, and then I will show you the results from the greenhouse and the field experiment. So going to the field experiment that we did, we had various plants such as squash, cabbage, broccoli, collards, and cantaloupe next to a central row of red giant mustard. So we had 4 rows per crop, and we took weekly leaf samples at 5 intervals of 2.4 meters within each row and every 1.5 meters in the mustard rows. And we then counted the number of adult and immature whiteflies after a certain period. And we counted 20 leaves per crop. So the results that we got showed that in the greenhouse, there were lower numbers of eggs that were laid on the giant red mustard or red giant mustard. The pairs of colored plants showed higher egg numbers than the pairs of red giant mustard plants, or the pair of the mustard and the colored plants. In the field experiment, we found the highest whitefly oviposition on the squash and the cantaloupe, and the lowest whiteflies were on the red giant mustard. And the other crops hosted intermediate numbers or intermediate densities. So we concluded then that in terms of the laboratory study of choice and no choice test, we found a reduced oviposition on squash plants that were sprayed with mustard and other oils. In the greenhouse, we found that the colored pears had higher oviposition than the mustard pears. In the field, the squash and cantaloupe had the highest numbers of whitefly. So this could possibly be used as trap crops because they are able to attract the target crop, the target pest away from the target crop. And finally, the intercropping the red giant mustard in the field may be a promising strategy to control this whitefly. So knowing that and getting that information, we went ahead. We have a 6-acre field near our laboratory, so I thought we could just go ahead and plant some native and native plants or weeds that would be good companion plants, as well as planting annual plants. And these companion plants can serve as a source of natural enemies because these the flowers have the nectar and the pollen as a food source for natural enemies that can then help reduce the target insect pest. And as well as during, for example, the summer months when there's no vegetables being planted or the field is just left fallow, then we would plant buckwheat as a cover crop and ryegrass during the winter season. So we sort of we did some practical applications based on the research that we found. Let me see, something is going on in my, I may have pressed something here. Okay. So part of the study I did with the push-pull is looking at the natural enemies, the population studies of predators such as hoverflies, lady beetles. These are good natural enemies or insects that attack target pests such as Bemisia. We could use refuge plants such as annuals, sweet alyssum, Queen Anne's lace, buckwheat, yarrow are good horticultural plants we could plant around the borders, as well as using perennials or even just weeds. For example, in Florida, blue mist, flower, lesser snakeroot, dotted horsemeat are all native weeds that could be used to help increase natural enemies. And an example of a predator shown here is the Delphastus catalinae, which is endemic. And I mentioned using cover crops such as buckwheat, ryegrass, or sun hemp to attract beneficials. And in addition to that, you could enhance the soil nutrients and reduce the weeds in your crop. As I mentioned, these attractive flowering plants can be used to attract hoverflies. For example, the picture below shows a hoverfly that could feed on sweet alyssum. For example, as shown here in the picture, in the top right, I had sweet alyssum planted right next to lettuce, for example. You can intercrop these so that it would attract the natural enemies. Other predators that could be useful are tachinid flies and parasitoid wasps. So these are just some samples of crop fields that we tested to look at the use of these companion plants. For example, the picture at the top left, we had sweet alyssum next to kale crop, and then the lower right shows broccoli planted next to sweet alyssum, a row of sweet alyssum. So going back now to the use of the push-pull technology and companion planting, these companion or insectary plants provide nectar or pollen to beneficial insects that then could attack the target insect pests. For example, in this case, the white flies that I showed earlier. There are commercial plant products, for example, Predilure, which is practically methyl salicylate, that have been commercially produced to attract natural enemies in the field or in your greenhouse. So I was interested to look at these attractiveness of sweet alyssum and methyl salicylate to hoverflies in vegetables. And I did a study in fall of 2014 and spring of 2015 using the Malaise traps, which I showed earlier. We conducted the study in a vineyard next to our labs, and we put potted sweet alyssum under these Malaise traps for 48 hours. And I had 4 treatments. One was the use of methyl salicylate alone or Predilure alone. The second treatment is just having sweet alyssum under the Malaise trap. And then the third is using the methyl salicylate and sweet alyssum. And then the fourth is the control, which we just left the trap empty. So in the spring of 2014, we used kale as the target crop, and the Malay strap is shown here on the right, where you put that over the potted sweet alyssum in the middle of the kale crop, and we have a collecting head up here, which shows, which is used to collect the insects. And then the treatment here was a trap with sweet alyssum and another with no sweet alyssum. And this is the data we collected during the time. In 2014, you can see in the x-axis from October to November of 2014. And then the y-axis shows the total number of hoverflies, or the insects that we collected from the Malay straps and the various treatments. So you can see here the red graph shows the population of the hoverflies in the treatment, where we combine both sweet alyssum and methyl salicylate, had the highest numbers of hoverflies, followed by just sweet alyssum, and the control shown here by the white line graph, and the lowest numbers are the control, where we didn't have use of any sweet alyssum or methyl salicylate. And then in spring of 2015, we conducted similar treatments in the field, and these are the population densities we collected from April of 2015 to May of 2015. And similar to the 2014 study, we also collected most of the total, most of the hoverflies in the treatment, where we had both sweet alyssum and methyl salicylate, followed by just alyssum here in the green line graph, followed by just the methyl salicylate and the control. And this is just the mean numbers of the hoverfly in this bar graph. So the numbers are shown on the left during the fall 2014 study, and the one on the right is the spring 2015 study. And you can see that highest numbers of the hoverflies were found in those where we used alyssum or sweet alyssum and methyl salicylate, compared to the control, or the ones that just had methyl salicylate. We also identified the different species of hoverflies that we collected from the Malay strap. So this pie graph shows the most abundant species. Here in green is the Toxomerus marginatus as the most abundant hoverfly that we collected, followed by these other species, Allograpta obliqua, Toxomerus geminatus, Toxomerus botzai, Pseudosaurus clavitus, and Toxomerus floralis. So this gives you a little indication of the types of species of hoverflies that we were collecting out in the kale field. And then in spring 2015, we also found very similar population densities, where Toxomerus marginatus was the most abundant hoverfly collected. And then, since we did find that this species, this Toxomerus marginatus, was the most abundant, we did a simple study where we just did a comparison of counts of hoverflies between a treatment where we just used sweet alyssum and a control, and that was in April of 2014. This was much earlier on than the ones that I showed previously, and you can see here the difference that those that had sweet alyssum had the highest numbers of marginatus through time, compared to the control where we did not have sweet alyssum as a companion crop. So to summarize the results, then we found that sweet alyssum with and without methyl salicylate attracted significantly more hoverflies. Toxomerus marginatus hoverfly was the most abundant out of the 6 or 7 species that I showed earlier. The kale plots with the sweet alyssum attracted the highest numbers of hoverfly, Toxomerus marginatus, and that sweet alyssum is an attractive refuge plant of predatory hoverflies. So these are just pictures or demonstration of the plants that we put out as a demonstration to our stakeholders. We planted annual and perennial plants in vegetable field plots. For example, here during the summer of 2017, we planted rows of flowering plants here right next to the crop. And again, on the right, you can see here also some of these commercial, it's a mix of various flowering plants right next to, we had okra and we had eggplant next to it. And then also next to our greenhouses, we had a demonstration of use of the native weeds, for example, can be used as attractive plants for not just the hoverflies, for example, but also the native pollinators, such as bumblebees, as shown here on the left. Here's another picture of just some of the commercial flowering plants that we would plant next to a crop. And this is sort of already at the time of harvest of the crop, so you don't see the crop very well. But on the right, you'll see some hot peppers, and next to it are the commercial flowering plants. And around our labs as well, we went ahead and also planted perennial weeds and flowering plants to increase and maintain the beneficial pollinators and natural biocontrol insects. And here's just another picture of those flowering plants around our laboratories. In 2017, I had an opportunity to be a Balik scientist through the Department of Science and Technology, and was hosted by Dr. Divina Amolin from De La Salle University. And I had a chance to work on the cacao pod borer to help control these pests in cacao plantations. So we went to Isabela State University and the Benguet State University, as shown here in the map. And I noticed when we got to the entrance of Isabela State University, there's an area there at the entrance where I would say maybe a quarter mile or maybe even maybe not a half mile, but along the road were cacao and coconut trees planted, as well as these flowering marigold zinnias that were planted all along the length of that road that leads to the university, the entrance. And so I thought that was interesting, and when I asked about it, that was the purpose. They planted these so that they would have this source of natural enemies to control the insect pests in cacao and the coconut right next to it. And I saw the same thing in Benguet State University. I just don't have a picture here, where they were also using these flowering plants for source of beneficial insects. And this is just another picture showing on the left the marigold and zinnias right next to the cacao plants. And the one on the right is just a picture of cacao pods, as well as the drying of these cacao seeds on the right. So I'd like to switch gears now and talk about my research at the Florida A&M Research and Extension Center using this push-pull technology and companion planting. In this case, we received funding from the USDA, USDA National Institute of Food and Agriculture to look at the use of high tunnels, or an example here, as shown in the picture on the right, it's mainly a screen enclosed structure, and try to see if this could be adapted for use in Florida, and that would help lengthen the growing season of vegetables that are planted in Florida, because it does get cold during the winter months from December, November, December to March. So having these enclosed structures or high tunnels will help in growing more vegetables during that period. The other types of high tunnels, which you may be familiar with, are the ones that open at the sides. And this would be during the summer, you could still be able to plant some of these cool season crops by using these high tunnels. And that was the purpose of this study with faculty at Florida A&M University, where we were testing the use of these tunnels, so you could still grow leafy vegetables, or for example, lettuce and other crops that are normally planted during the cool season, but we could be able to plant during the summer season. So we tested it in 2018, where we had mustard in this enclosed structure, and other leafy green vegetables. And right next to it, we just had buckwheat as a cover crop to serve as a source of natural enemies, and also to help with increasing nutrients in the soil. On the right are my collaborators with Dr. Alex Balquez, and also his student and technician. So the idea for push-pull technology, I sort of made an infographic on it to explain it a little bit more, is that we push away the pest away from the target crop, as shown here on the left, using repellent plants or plant products, repellent plant products which have volatiles that push away the pest away from your target crop. For example, here is tomato, and then you pull them in, or you pull the pest away from that target crop and pull them into a trap crop, for example. And there are volatiles that are coming out of the trap crop that could then attract these insect pests. And at the same time, we use biocontrol, or the use of these good bugs against these insect pests or bad bugs. So in this case, I used the whitefly as a target pest, and using ladybugs here, for example, as the good bugs that could attack the whitefly pest. So this is just an infographic to explain it a little better. So in 2019, we did a study on tomato crop inside these top vented high tunnel. And we looked at using marigold, which we planted on the sides of the enclosure. And we also used African basil, potted African basil plants as a companion planting. And this was during summer, so marigold would grow well during this time. The African basil, I didn't realize, could pretty much be a perennial. And in this case, we mainly used potted plants so that we could move them around the enclosure as well. So on the top right, on the right, you'll see potted plants of the African basil, and next to it are marigold that we planted on the edge of the enclosure. And then on the bottom, left of the screen are some of the repellent plants that I tested, or push plants, such as citronella from the left, society garlic, and lemongrass. And this is the data that we collected during that season from June to November of 2019. And the main insect pests in tomatoes were whitefly, aphids, and thrips. These are the three main ones. And so you can see here the black line graph here is the whitefly. So you can see the higher numbers of whitefly compared to the other pests, such as the aphids in the red line graph here, and the thrips is the green line. And there's the purple lines, the black unidentified thrips. In terms of natural enemies or the good bugs against whitefly, for example, aphids and thrips, we identified two parasites, Eretmocerus species and Encarsia species were present during that time. So this dual strategy of push-pull and companion planting, it seems to be a doable strategy where we can use repellent plants to push away the pests. For example, here on the right top is the thrips, and next to that below is the aphids. And then some of the good bugs that we collected, or the natural enemies that attack the whitefly, for example, aphids and thrips, are these minute pirate bugs. And then in terms of the beneficials, as I mentioned, the whitefly parasites were also good beneficial insects. So the pull component here in this strategy, we were going to test green leaf volatiles such as leaf acetate and leaf alcohol. But during that time, the numbers of whitefly and pests were just so low, we couldn't really test them. But we continued to test them in the laboratory. For example, these leaf acetate and leaf alcohol were showing promise as a pull component in this technology here. And so we continue to do that study. My technician continues to do that, or my former technician continues to do that study in the laboratory and in the greenhouse. And then I mentioned the beneficial refuge for flowering plants, and we did include Predalure in those potted plants of African basil. So after we did the weekly samples of the whiteflies, the pests, and the beneficials, using sticky traps and leaf counts, we found that we found low numbers of sweet potato whiteflies, aphids, and thrips, as well as yellow striped armyworm as pests in this enclosed structure. And these were in comparison with numbers that my collaborators were also testing in their high tunnels in Florida at the Citra, and as well as my collaborators in Georgia as well. They were finding higher numbers of these whiteflies, aphids, and thrips. But after I presented to them the results of these studies, then they also started using this push or repellent plants in their high tunnels. And they did observe that the numbers were also decreasing, the numbers of pests. In January of 2020, we went ahead and did a study on leafy greens. So that was in the same greenhouse in Quincy, Florida. You can see on the left, we had marigold planted along the side of the enclosure, followed by a row of sweet alyssum. And then you can see the different varieties of leafy greens that we had planted in in this screen enclosure. The repellent plants that I used here are our Society garlic, mustard, the variety Caliente and red giant mustard. And I also tested the arugula variety Niman. And we intercrop these with the leafy greens. And as I mentioned earlier, we just used potted plants so we can like move them around the field or around the plots. This is another picture of that, the leafy greens inside the screen enclosure, and then the African basil are shown here on the left bottom picture. The white packets are that you can see on a bamboo stake there that we just inserted in the pot were the Predilure packets, and then we use these yellow sticky cards to collect the insects. And these are the numbers that we found through time. We put out 12 sticky traps, and these are the numbers of insects that we collected from 12 traps from December of 2019 to March of 2020. And you may have noticed this is just right before the pandemic, and we were shut down by then in March. So this shows that aphid counts were much higher in the leafy greens, followed by the thrips and the whitefly, and the two parasites, the Erythrocerus and then Carcia, were the two main parasites that we collected at that time. So again, to summarize this dual strategy of push-pull technology, companion planting using repellent plants such as mustard, arugula, and Society garlic, shown here. And the pull component, as I mentioned, we were trying to test was the green leaf volatiles. So it's still an ongoing study using an olfactometer. And the companion flowering plants we used were basil, marigold, sweet alyssa, as well as the commercial Predilure packets to attract the beneficials. And the results were mainly a low population of these whiteflies, and the natural enemies were mainly the whitefly parasites. So this dual strategy seems to be a low-cost, environmentally friendly pest management tool to control insect pests. And I'd just like to acknowledge some of the folks that helped me conduct this study, mainly technicians and students at Florida A&M University. And my technicians and my collaborators, Dr. Balquez and my collaborator from UF, and Dr. Xin Zhao, and this was funded by USDA NIFA, as I mentioned earlier. So I'd like to stop there. I was also keeping track of time. I didn't want to spend too long, and I'd be happy to take questions.
Thank you very much, Dr. Jesusa Silegaspi, for that very informative and comprehensive presentation on the topic push-pull technology and companion planting, a dual strategy for insect pest management. So now, we will proceed to the open forum. May I now call on Dr. Regino for our Q&A moderator. Sir, thank you.
Okay, thank you very much, Ma'am Dara, for the introduction. And of course, thank you, Dr. Jesusa Silegaspi, our speaker, for your very comprehensive talk and for sharing your knowledge, expertise, and insights on the push-pull technology. Now we will open the floor for the question and answer portion. I would like to invite our participants to type their questions in the chat box or use the raise hand function if you wish to directly ask the resource person. Once called, please state your name and your organization. To our attendees watching on our College of Agriculture and Food Science Facebook page, just type your questions on the comment section so we can relay them to Dr. Legaspi. Okay. So we have Dr. Legaspi, we have a question here. Our first question comes from Mr. Bernard Panabang. So this is his question. So there is this field practice that mustard is used as sort of disinfectant in the field by planting them in bacterial wheat infected areas, and you can't grow other brassicas right after mustards. I was just wondering whether it would have an effect on the yield of other crops. It is intercrop with, or say, a sort of an allelophatic effect on other crops. So Mr. Bernard is asking if there is this effect on the yield of other crops if we will intercrop this type of plant with other crops. Is there an allelophatic effect, ma'am?
I didn't really specifically look at the allelopathic effect. From the study that we conducted in the screen enclosure, my collaborator, he's a horticulturist, and his interest was finding out the yield from the different crops that we planted. And it seemed from his data that the productivity or the yield from the different varieties of target crops that he planted were in line with the yield that you would expect if you planted them conventionally or in a standard practice. So I'm not sure that would be a good way to explain whether there may be allelopathic effect or not, but it seemed like in the study that we conducted, the yield was pretty much the same as what we would expect in using standard crop production practices. It is possible that there would be a very interesting to study that perhaps in the future to look at the effect of allelopathy.
Okay. So Mr. Bernard Panabang, so Dr. Legaspi is willing to study this type of the question that you ask. Okay. Another question, Mom. Our next question comes from Ma'am Rochelle Soriano. Good morning. May I ask the possible cover crops that can be used under Philippine setting?
Well, many of these plants, I'm pretty sure, because of the weather we have in Florida, especially in South Florida, is very comparable to the climate, sort of a subtropical region in the Philippines. So I would say some of these companion crops or cover crops could be useful. Could be planted. For example, I mentioned also that the pictures I showed in Isabela and Benguet, many of those, for example, are crops or plants that we could plant also here in Florida. So I would say that many of these are relevant to be or comparable to what could be planted in the Philippines.
In relation to this, Ma'am, there is this question about the cogon grass. Are you familiar with that, ma'am? Cogon grass. Can we use that one as a cover crop?
I can't really say for sure because I know that sometimes cogon grass can be an invasive, for example, here in the US, it's considered an invasive weed just because of the life history of this weed. So I really cannot say that I could probably, probably follow that up later. But for now, I couldn't really say if that would be a good cover crop.
Okay. So we have another question, ma'am. So can you provide your perspective on this? So one anonymous attendee is wondering whether you have any recommendations as to the right combination of crops and flowering annuals with the focus on producing the pinakbet vegetables, specifically eggplant, okra, sweet pepper, bitter gourd, tomatoes, sitaw, and
Yeah.
squash. Okay.
Right. As I showed in the pictures earlier of our plantings of eggplant, okra, and that's during the summer time here in Florida, sometime in, I would say June, July. You could plant those commercial flowering plants as well. Many of them, I would think you should be able to plant there. For example, cosmos, snapdragon. I'm pretty sure these are common flowering plants as well. Marigold. Those can all be planted during the hot summer months, even in the Philippines. So I would suggest even basil, I think would be a good companion crop or plant next to your okra or those types of crops. Sunflower, those types could be planted next to those vegetable plants.
So we can use basil, sunflower, marigold
Right. Yeah.
as companion crop. Okay, ma'am. So another anonymous attendee is very much interested in this technology, and he is asking if there are specifics or if there is a specific methodology used in data gathering for the insects. Is there any specific distance requirement in conducting these types of studies?
It would really be dependent on how big your plots are in your study. So, for example, our row, the distance of our rows are only like 100 feet long, and about 6 feet between rows. So, in terms of the methods for sampling, I can provide copies of some of the papers that we've already published. But again, it depends on how the size of your experimental plots and the numbers of replicates that you would like to use per treatment.
Okay, thank you, ma'am, for that answer. So we have another question from an anonymous attendee. Can insect pests also have access to these non-prey resources such as pollen, nectar from flowering plants? If yes, how can we make sure that only the natural enemies, such as hoverflies, will be benefited from these resources, and not the insect pests, which are also present in the farm? Insect pests can also feed on pollen and nectar generally.
Yes, that is true, that they are also feeding on this pollen and nectar. Most of these, again, most of these insect pests are feeding on the leaves of the plants or the stem or whatever is in particular that they're feeding on that allows them to populate the plants. And so having the purpose of having these flowering plants were really to be able to have a source of natural enemies at the same time that the insect pests are found in the field. Previous studies have shown that the predators and the parasites usually come much later than the time that the insect pest is there. So most of the time, by the time the insect pest could be controlled, it's pretty much too late already because the pest had a head start much earlier than when the predators and the parasites arrived. So the main purpose of having those sources of plants, flowering plants, were to be able to have these source of natural enemies at the right time when these insect pests are just building up their population in the target crop.
Okay, thank you, ma'am. So we have here another question, ma'am. So one attendee is wondering if the push-pull strategy is suitable for use in countries with relatively small fields, such as Taiwan and Japan.
I would suspect that it would still be feasible to use this technology, partly because the areas where I actually did the study were in either organic growers fields, which are pretty small acreage compared to the conventional commodity growers that grow, for example, cotton or corn or tomatoes. So I would suspect that they would still work well. The nice thing to consider or to appreciate is that the diversity in around the target crop, and the more diverse it is, which is what you find in organic production, it will be better in terms of keeping the population of natural enemies in the plots or in the target plants.
Okay, thank you, ma'am. Now we have these questions specific for a particular crop, ma'am. Is there a push-pull technology for rice?
I would think not particularly that I've come across. There has been, I know, success, for example, in Africa, but not in rice, but in other crops. I don't really recall much studies that have been done in rice. But it doesn't mean we can't do it. I think it would be interesting to get more information about it. Perhaps more in the upland rice or non-flooded rice. It may be doable because I know, for example, in some parts in the Philippines, they do plant vegetables right next to upland rice.
Okay, so push-pull technology is also doable for rice.
Yes.
Then another question, ma'am. I think this is from a foreign attendee. So except using plants as the push and pull components, do you think some of the LED illumination devices may also be considered as the push-pull candidates?
I suppose if you could I think as I mentioned in that infographic that I showed, is that, the main idea is just finding a way to push the pest away from the target crop, and whether it's going to be use of light, use of LED, or use of other um, materials. I would say, yeah, you could use this as a push strategy, for example, to push away the insect pests. At the same time, you could also use light to attract, for example, when we're trying to catch insects at night, we normally would use a light trap to attract insects. So that I would say would also be possible
Okay.
part
So,
of the strategy.
okay, thank you, ma'am. So to Miss Shoyi Wang, so it is possible to use of lead illumination devices as push and pull candidates also. Okay, another foreign attendee, ma'am, from Rahan Nepal Agroforestry Polytechnic Institute. So he's asking what research innovations are currently being pursued to further improve push-pull technology for pest management.
I think I recently just did a review of use of push-pull technology in agriculture and forestry as well, but this hasn't been published yet. It's in press, but from my understanding, the push-pull I would think is going to be really very useful, for example, in developing countries, compared to developed countries, mainly because of the labor cost is quite can be prohibitive for using this technology, in big commodity crops or big acreage crops. So, anytime we could be able to reduce these labor costs, I think would be innovative if we could find a way to be able to still use this strategy, but with less labor cost.
Okay, ma'am. Thank you po. So, ma'am, indeed, we have a very interesting topic because we have a lot of questions, ma'am. Another one from Mr. Bernard Panabang. He's wondering whether you have studies for below ground pests, such as nematodes and other solid burrowing arthropods, and the effect of using push-pull strategy on soil health. About soil health, no?
Yes, that's quite interesting question. It's something that I've been interested myself. We did a study probably 5-6 years ago in Sunham, and I was curious to find out how the pests in the soil or under the soil are affected using Sunham as a cover crop, which is being encouraged here in Florida, in South Florida particularly. And what we were finding from that study, actually a publication will come out soon on this study of these underground pests, natural enemies that we collected under the soil in Sunham. And we were finding really good numbers of these tiger beetles, which are good predators. And these are good predators of insect pests in crops such as Sunham. And yes, I would think that it would be interesting to look at that angle there. Not particularly, at the time when we were doing the study, was to they use the push-pull method as well as the companion planting. But I think that would be something that would be interesting to look at in the future, and you could actually correlate the natural enemies that you are collecting under the soil with your insect pests in the particular crop that you're studying.
Okay, thank you, ma'am. So we're done talking about the advantages of push-pull technology. Now, ma'am, there's this question about the disadvantage of this technology. Is there any, ma'am?
Well, as I mentioned, one was the labor cost. That's why I think it hasn't really taken off. It's been done. I mean, this is not a new technology. It's been done, for decades previously in other, a lot of them from developing countries that have been using this method. But again, as I mentioned, the disadvantage is that it is very labor intensive, as I showed you even in that just one enclosure that we were looking at. It's not the type of planting where you could use tractors, all these mechanized production methods. There was studies that were conducted in California where they were using sweet alyssum intercrop with lettuce, and they were using mechanized production. So there is that possibility, but again, the disadvantage there would be the labor costs, as well as farmers that prefer, to get a profit from their target crop rather than using that, parts of their acreage to companion plants where they will not really make a profit from the yield of these types of plants. So because they're not cash crops that you're planting, so that takes away a little bit of the profit for the grower. So those are just some of the disadvantages of using this technology.
Okay, thank you, ma'am, for answering that question. Another one, ma'am, another question. What are the insect pests that can be trapped in Malay traps? Can this trap be improved using locally available materials?
Yes, the Malay trap actually is sort of a trap that is modified from a trap that I was using in the 70s in the Philippines. We were calling that at the time emergence traps, but it's very, very similar. And these are all materials I would say you could pretty much get locally. You can get organza type of netting, and you can pretty much sew these types of trap with local people. Yes, they can be modified in such a way that they can use local materials.
Okay, so really we can use this one in Philippine setting.
Yes, yeah, and then I think the other question was what type of insect pests that you could collect. Pretty much any kind of insect that would get caught in the trap, and using the behavior of the insect, where an insect normally move upwards, and that's why we're able to collect them in the collecting head, because of the behavior of the insect. As long as the insect gets caught in the trap and moves up in the collecting head, it can be caught. So at that time, but we were using those traps in rice in the Philippines. We were collecting the insect pests in rice, such as the plant hoppers, but we would also collect others such as moths, fall armyworm, and others. So using just the behavior of the insect, or knowing the behavior of the insect, you should be able to collect the target insect pests that you're trying to collect.
Okay, thank you, ma'am. I think you already mentioned this one, but let me just again ask this question. From Mr. Jomar Osoteo, how to prevent the strips and whiteflies in tomatoes? Maybe he's asking, aside from push-pull, or do you have any other recommendations, ma'am?
Yes, for aphids and thrips, I guess similar to whitefly, we should be, if you're trying to find a non-chemical type of control, you can use what we call safer soap, or it's pretty much really just drops of dishwashing liquid with water, and you can spray that on your plants to help control your insect pests. So it's a non-chemical way of controlling aphids, for example, and thrips.
Okay, thank you, ma'am. Another question from Miss Angel Lee Alcalde. Can this technology be applied in hydroponic culture?
Yes, yeah. Thank you so much for that question, because it's something I've always been curious about, because I was involved in projects where they were looking at hydroponics, and I actually also visited several greenhouses that were run by big companies that used hydroponic system for vegetable production. Pretty much, I have not seen this method used. And I think part of the reason is that, it's the hydroponics that they've done or used here in the US have been in enclosed structures. For example, like huge greenhouses, which pretty much, I think people think because it's so enclosed and protected that they will not get the insect pests or, have the beneficials inside. But as you can see, even in the enclosed screen structure that I showed, we were collecting all these various insect pests and natural enemies inside. So I have not really seen this technology used in hydroponics. It doesn't mean it's not, but I just haven't come across documentation of it.
So maybe we can have further research on the use of push-pull technology in hydroponics culture.
Yes, for sure.
From our Facebook page, ma'am, there is this question from Miss Richie Soriano. Is it okay to just leave the cover crop in the area, even if you already harvest the main crop?
You could leave the cover crop in the area, although in some cases, leaving the cover crop may cause some other unintended consequences, where you may be attracting other types of insect pests or other types of others, that you may not have intended to be there. So, but I also don't see a reason for removing the cover crop if they're not already dried up, for example, because it just has already reached its maturity stage. So I don't really see also a reason to have to harvest the cover crop if you don't need to harvest it.
Okay.
Most people would harvest because they need to use the land for other purposes. after the cover crop has already matured and dried up.
Okay.
Mhm.
Thank you, ma'am. Maybe we'll have two more questions, ma'am.
All right, thank you.
Okay, and one is from Joseph Mordido. So his question is so I'll read his statement here. In the Philippines, we have 1 million hectares of coconut that have some affected of diseases by insect pests. Will push-pull technology help solve the problem of the cocoa farming in the Philippines? And maybe you can cite your recommendations, ma'am, that could be used in this technology for our farmers, cocoa farmers.
Yes. Well, I can't really say for sure what types of particular push plants or repellent plants. It's something that we were interested to look at, and I think sometime in the future we'd be able to look at that in collaboration with faculty. I think Dr. Divina Amali may be in the audience, and we have been discussing with other faculty from Florida A&M University and other agencies actually in the US to look at using the push-pull technology for pests. For example, red palm weevil is a big problem now, I know, in coconut in the Philippines, as well as the coconut rhinoceros beetle. So we do have a proposal to look at that, and hopefully, if it does get funded, then we'll be able to collect some data to find out if this technology would be useful in coconut against insect pests, such as I mentioned, the red palm weevil and the coconut rhinoceros beetle. But for now, I really couldn't say what has been successful using this technology against the coconut insect pests.
Okay, ma'am. I think we can end the Q&A there, ma'am. That for those, actually, ma'am, we still have more questions, but due to time limitations, so for those attendees who were not able to answer your questions today, okay, please do not hesitate to contact our experts through our Facebook page by sending a message there, UPLB College of Agriculture and Food Science. Okay. Again, thank you very much, Dr. Legaspi. Indeed, we appreciate your effort for taking the time to be our speaker for today's webinar session. We also thank you for your efforts in explaining some of the practical aspects of implementing the push-pull strategy. Okay? Your contribution has deepened our understanding of this important aspect of sustainable agriculture. And we look forward to further collaboration in the future. Okay. And of course, thank you to our attendees for your engaging questions and for your active participation in our discussion. Again, if we were unable to answer your questions, please do not hesitate to contact us by sending a message to our Facebook page. Okay, so this concludes the question and answer session. We hope you found this discussion informative and insightful. Now I will hand the floor back to Ma'am Dara. Ma'am Dara, it's all yours again.
Thank you. Thank you, Dr. Nino, for administering or moderating our Q&A. And thank you very much, Dr. Legaspi, for addressing all the questions and clarifications. So let us now proceed with the awarding of the Certificate of Certification to Dr. Legaspi for sharing her expertise for us today. So let me read lang po the citation. College of Agriculture and Food Science, University of the Los Baños, Laguna, Philippines, present this certificate of appreciation to Dr. Jesusa C. Legaspi for serving as the resource person on the webinar entitled Push-Pull Technology and Companion Planting, a Dual Strategy for Insect Pest Management during the Plant Health Webinar Series held on October 19, 2023 via Zoom, given this 19th day of October 2023 at the College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna, Philippines, signed by Dr. Elpidio M. Agpisit Jr. Thank you very much, Dr. Legaspi.
Thank you so much for the certificate. I truly appreciate it. And thank you for the invitation to present my research here and being able to interact with you all. It's been very productive and fulfilling, and I hope has been very helpful and providing information about this strategy. Thank you.
Thank you very much, po. With this, po, we hope to see you all again to our next webinar series. Brewing soon is the CAPS Coffee webinar series. To receive updates or details about it and of our coming events, follow us on Facebook and subscribe to our YouTube channel of the College of Agriculture and Food Science. If you would like to receive an e-certificate, please answer the evaluation form that you may access it through the link in the comment section or may scan the evaluation link already posted on your screen or shown on your screen. Thank you for attending this episode. Now, we are almost at the end of our program. Therefore, may I call in the director of the Institute of Food Science and Technology, Dr. Dennis Marvin O. Santiago, for his closing remarks.
Thank you, Ms. Dara. Okay, am I audible?
Yes, sir.
Okay. Thank you very much. And for today's webinar, I don't know if this will be the last for the plant health series, but thank you very much to Dr. Jesusa Legaspi for the very informative presentation of her research as a non-plant pathologist or entomologist. I really had a good time and learning from your presentation. So I think my key takeaways is that we can use plants as to push away the pest, and also we can also use plants to attract the good insects, and then they will be used as a biocontrol for the pest. So I think that will be a good strategy for our farmers to be used in their plantation. And I think as a food technologist, the main benefit that I see from here is that the produce of these plants will be beneficial in terms of there will be greater yield for the produce. And then, of course, the quality of the fruits or the different vegetables that will be used will be of standard for food processing. So I think that will be the main benefit of this technology. And thank you, Dr. Legaspi, for presenting this. And then I hope our participants here will be able to use this kind of technology. And maybe on your own, you can do that as a practice in your farm on how we will be able to take advantage of the plant species that can be used to push the pest away and pull them into some trap crops, and then will be used for and the other flowering plants to be used to attract the beneficial insects. So I think with that, we had a good time today, and I hope you will be able to attend again. I think there's something brewing in CAPS for the another series for the coffee series. So I hope you will be able to attend that. And with that, from the CAPS family, so Hiraya Manawari, and take care, everyone.
Thank you.
Thank you very much, Dr. Santiago. Once again, thank you, Dr. Legaspi. For those asking, yes, you will all receive an e-certificate. Just kindly answer the evaluation form that you may access by scanning the evaluation link or going to the link posted shown on the screen. Again, we would like to thank and congratulate the persons behind this webinar series. Thanks to the webinar committee and of course to all the participants for joining us today. We hope to see you on the new CAPS webinar series. Help us share our upcoming webinars and events. This ends the program. Again, I am Dara Maria Efraelin. Thank you for joining us. God bless you and have a great, even though it's a rainy day. Thank you.